Okay, the cell cycle. So we move onwards with that. So not a whole lot again to discuss on gap 1, basically chapter 9. Everything we learned in chapter 9, all the energetic stuff goes into the gap 1. The formation, the storage of ATP to help fuel then our very active S phase. So now let's focus on interphase, S phase, DNA synthesis phase. So I hope I don't have to go back and review DNA, right, from chapter 4. DNA, that very dynamic, very centralized, core dogmatic molecule that we learned and you should be experts on already. So we know that it's a double helical structure, sugar phosphate backbone, anti-parallel strands going again in opposite directions from 5 to 3 one way, 5 to 3 the other way. Adenine binds with thymine, guanine with cytosine, so you know all that stuff, right? And so, but you have to visualize what the molecule looks like for the S stage to kind of make sense completely here. So if we kind of unravel that double helical structure and we show it like a ladder, we remember that the 2 separate strands are actually held together by these little weak hydrogen bonds. 2 hydrogen bonds between adenine and thymine, 3 hydrogen bonds between guanine and cytosine. So these bonds shown in little red dots are the bonds that we're going to kind of focus on in our discussion here. So when the cell has to divide, when the cell has to, I should say not divide, when the cell has to replicate its DNA, it's going to do so in the most efficient manner possible. And the most efficient manner possible is what we're going to call semi-conservative DNA replication. Semi-conservative. Saving, if we conserve water, you can save something, you save. So we're going to save half. Half is saved, semi-conservative. So we're going to use basically each parent strand as a template for a new strand. So if you can visualize this picture here, we have 2 of the parent strands in blue. After we're done, we have utilized 1 blue strand and made 1 new red strand, saved 1 blue strand and made 1 new red strand. So we saved half of the original blue strands and we make new red strands that are complementary matches to that original blue. So kind of a very simplistic way of viewing this. We're going to look at more details here. So if we visualize then our DNA in blue, and we see then that we're breaking hydrogen bonds. And once we break hydrogen bonds, then all we have to do is form 1 new strand. Piece together 1 new strand, not in red now, this one is in yellow, but we end up with A with T, and over here we have A with T. On the left, C with G, on the right, C with G. So since we broke that complementary match, we're able then to make 2 new complementary matches, and then when we're said and done with everything, they're going to be the exact same relative to each other. So we are, again, most efficiently making 2 strands out of 1. So that's, again, semi-conservative DNA replication happens in the S stage of interphase. So this is the what, right? The how is a little bit more intricate here. So if you learn anything from chapter 9, we know that things happen in the pathway sequence, and pathways involve a lot of enzymes. Well, this is no different, right? There's a lot of enzymes involved here. We're going to look at our helicase enzyme, topoisomerase, primase, polymerase III, polymerase I, and DNA ligase. So these are the enzymes and roughly the way that they work. There's some overlap in the sequence of their actions. But I do want you to know these enzymes, they are relevant, they are very critical, and they help ensure that we have no genetic mistakes, no genetic mutations during this replicative process. So helicase, topoisomerase, primase, polymerase III, polymerase I, and ligase. So first, right, we have our double helix. That double helix must be open. It must be sort of torn apart. So this process begins with the help of helicase, and helicase has a pretty easy job. All it has to do is break hydrogen bonds. Those little hydrogen bonds that are holding the A's with the T's and the C's with the G's. So if we can break these hydrogen bonds, then we sort of unravel, we open up that sequence, that strand. Right? So I'm going to back up. So we show that yes, DNA has been split apart. Well, what split it apart? That would be helicase. Helicase is opening the helix, right? DNA serves as the substrate, and it's basically breaking apart these hydrogen bonds. So kind of like a little zipper is kind of zipping up, opening all of those hydrogen bonds to separate out these two strands of DNA. So with helicase, helicase can't necessarily function by itself. It needs the positioning, it needs help of this secondary enzyme we call topoisomerase. So if you can imagine a karate student, they're gonna break a board or something. Well, we need something or somebody to hold that board in place so that the karate person can chop it in half, right? So that's kind of what topoisomerase does. It helps position the hydrogen bonds so that helicase can come in and break those hydrogen bonds. So it's a collaborative effort between helicase and topoisomerase. So in this situation, we have yellow strand, which represents DNA, yellow strand, which represents DNA, and yellow strand, which represents DNA. So in this particular situation, we know that helicase would be right there at that Y, and it's working from left to right, unzipping the DNA, breaking hydrogen bonds, creating these little replication forks, as they call it. Look like little bubbles, little replication forks, because remember there's not just going to be one. It doesn't start at one end and works all the way across. Anywhere in that double helical structure in the middle, it can start in the middle. We can have two of them, and they start to work in opposite directions. So this speeds up the process. That's what enzymes do. They speed up the reaction, the process. So they're going to speed the breaking so that we can speed up the formation of two new strands. So again, helicase works with topoisomerase. Topoisomerase travels ahead of helicase in the replication fork, positions the hydrogen bonds, it prevents tangling, and makes it very efficient for helicase to break forward. So up to this point, with those two enzymes, we now have the DNA open. Okay, so we got to start now semi-conservatively replicating these. And that's going to be initiated by primase. So if you think of the word primer in Spanish, first, prime, if you're going to paint, you're going to prime something first. If you don't paint it red or blue first, you paint it white. You prime it, and then you paint with the actual color afterwards. So we open the strand, and that first sort of anabolic process that begins now is going to be initiated by the primase enzyme. It's a little bit weird. It will place not a DNA primer, but an RNA primer. So again, we're trying to replicate DNA, but we're starting with an RNA primer. A little bit odd, but if you remember, RNA is a little bit of a simpler nucleotide, very similar, but again, single strand, and that's why we start with this here. So we place an RNA primer to initiate replication, which is that little primer is just a short section of RNA. So in blue, we have our template, our DNA that's been split apart by helicase. And now this little green weird shape represents our primase, our enzyme. This is critical. all of these replication enzymes work in the 5-3 direction. So they're going to work in the 5-3 direction. So they have to have the DNA positioned in the 3-5 direction. So if you notice in blue, we have 3-5. That's the correct position that must be in place in order for us to start priming. So now we start priming in the 5-3 direction. So we can prime from left to right in this situation here. Using DNA as a template, Primase starts to add a small RNA primer in the 5-3 direction. Okay, so the primer did its job. Primes a little section, and then now it kind of, okay, I got it started, now you all take over. So enter now DNA polymerase III. And again, the numbers are weird. The numbers don't work, they don't operate in the sequence that they're named. They were named in the sequence in which they were discovered. So this one was discovered first, and then another one was discovered in bacteria, polymerase II in prokaryotic cells, and then a third one, polymerase III in eukaryotic cells. So they were discovered in a different sequence than what they actually function. So polymerase III actually works before polymerase I. And polymerase II only works in prokaryotic cells. We're not dealing with prokaryotics, we're dealing with eukaryotic cells now. So DNA polymerase III follows that little primer and adds new DNA bases in the 5-3 direction. So it makes a polymer of DNA in the 5-3 direction. After it does what it needs to do, we go then back to polymerase I. DNA polymerase I goes back and removes the RNA primers and inserts actual DNA. So it replaces DNA with that old original RNA. RNA is removed, DNA is put in its place. And then lastly, but not least, we have DNA ligase. DNA ligase is what actually forms the bonds between the phosphates and deoxyribose. So remember, all we're doing here is we're adding the nucleotides, right? They're connecting our, basically our hydrogen bonds. But we got to make sure that those phosphodiester linkages are set in place there, right? Our phosphate and sugar backbone has to be correct and in place. So think of DNA ligase like that final inspector. It's the last chance that we can, go and analyze and check to see if everything was replicated correctly. So once DNA ligase approves, then that DNA strand is considered complete and sent out where it needs to go. So to kind of give you a visual here, right? We had left off with our 3-5 DNA template. We start the priming with primase, a little short green little RNA primer. And then that enzyme leaves, we kick it out and enter now this little donut looking thing, DNA polymerase III. So DNA polymerase III arrives and starts to add DNA bases in red, the 5-3 direction. And once it gets there, polymerase III is done. So enter now polymerase I to come remove that green primer, replace it with red DNA, and then that last DNA ligase to make sure everything is correct. Now I'll put a video up to help you kind of visualize this in action. Something that's going to be a little bit not difficult, but just a little bit tedious at this point is that we're going to have two strands, and we're going to call them a leading strand and a lagging strand. And the reason behind this is, again, because we can only work in the 3-5 direction. The template has to be 3-5. DNA can only be added in the 5-3 direction here. Yeah, so we only can work one way. It's like your car that only works in drive but not reverse. So it only goes one direction and doesn't go backwards. So we're going to have two strands that have to be built in 5-3 direction, but they're going to be formed in opposite directions. So one of them is going to take longer, one of them is going to be faster. So one is going to be leading the race, one is going to be lagging behind. So let's look at the blue strands here, right? So I had the original 3 to 5, this top strand, but the original DNA strand that was associated with that is 5 to 3. So again, if I'm priming and using my replication enzymes, sure, this is no problem. I can work from left to right in the 5 to 3 direction in red, right? So that's what we're trying to do. As long as Helicase keeps opening, we continue to follow in the 5 to 3 direction, no problem, right? Where we run into a little lagging problem is in this second strand, right? Because we're going 3 to 5 on the bottom strand, we're going from right to left 3 to 5, right? 3 to 5. And if you remember, we can only work in the 5 to 3 direction. So we have to prime, and then we don't work from left to right. We're going to work actually from right to left. So as we continue to open, this strand continues to win the race. It's going to be the leading strand, leading strand, leading strand. As long as we keep opening, this thing keeps replicating. The bottom strand, we have to prime and work. We have to prime again, work to the left, prime again, work to the left, and it's going to be formed in little fragments. So it's going to take longer. It's going to lag behind. And these little fragments are they were discovered by a famous geneticist, David Okazaki. So they're given the name Okazaki fragments, and the lagging strand only. Right? So lagging strand has Okazaki fragments, and I hope you understand why. Why do we have to build DNA in little fragments and not in a continuous fashion like what we have on this top strand there? So again, just the mechanics of the enzymes, they only work in one direction from 5 to 3. So to put everything together, we have then our top leading strand, 3 to 5, and it doesn't always have to be the top one. It just depends on how we're looking at it. But in this case, 3 to 5 on the top. So this becomes our leading strand. Primase put down that primer. Polymerase III is adding new DNA bases. Right over here we have topoisomerase that's helping to position, and here we have our helicase that's tearing apart that strand. We have our primase. Right, there's our primase that starts to work in the left to right situation. I should say, I'm sorry, primase in right to left situation here. So the primer, we prime and we work to the left. We prime, we work to the left. So it's just same process, just more work. After polymerase 3 has worked, polymerase 1 has to come in, remove the primer, replace with DNA, and then DNA ligase would come in and make sure everything is correct. So that is basically all that's happening in the S phase of interphase, right? So you can imagine that's taking up a lot of energy, a lot of ATP is being utilized. So after we burn all that ATP, yeah, we make DNA, but we burn ATP. So once we burn all ATP, we make all that DNA, we have to shift back to another resting phase, another G2 phase. So it's not as long as the first one, but it does the same job. It builds up more ATP to get us ready for another energy expensive phase that is mitosis. So with that, let me stop this one here, and I'll upload these with some sort of complementary videos that hopefully they'll help a little bit, and I'll continue with mitosis.